Hello everyone. Today we're going to be going over cells, the working units of life. So let's get started here. Let me just get my stuff ready real quick. Okay. So the key concepts are cells provide compartments for biochemical reactions. Prokaryotic cells do not have a nucleus. Eukaryotic cells have a nucleus and other membrane-bound compartments. The cytoskeleton provides strength and movement, and extracellular structures provide support and protection for cells and tissues. So our overarching question is, what do the characteristics of modern cells indicate about how the first cells originated? So cell theory was the first unifying theory of biologies. So cells are the fundamental units of life. All organisms are composed of cells. All cells come from pre-existing cells. Important implications of cell theory. Studying cell biology is the same as studying life, because they all have cells. Life is continuous, all the way back to the evolution of the first living cells. So life begets life. So first cell formed, supraprimordial goo, led to trees and animals, and little frog. Okay, so led to trees and animals, so it goes all the way back to the first cell. Most cells are tiny in order to maintain a good surface area to volume ratio. The volume of a cell determines its metabolic activity per unit of time. The surface area of a cell determines the amount of substances that can enter or leave the cell. So if you have a tiny cell, it's really easy to get to the center super quick. But if you have a big cell, it takes a lot longer to get to the center from any side. So this has a lot more surface area to its volume. This has a lot less surface area to its volume because it could fit like a ton inside of it. So what our bodies do is we have a whole bunch of cells that fill up that same space to keep the surface area to volume ratio small. So here we have a scale of the scale of life pretty much. So these are things we can't see, things we can see, and things that we can see just with our normal eyes. So atoms are 0.1 nanometers, small molecules are a little less than 1 nanometer. Lipids are a bit bigger than 1 nanometer. Proteins can be up to 10 nanometers. The T4 phage virus is about 100 nanometers, which is still way tinier than a cell. The chloroplast, which is inside of a cell, but it's about the same size as a prokaryotic cell, is at the 100 nanometer to 1 micrometer mark. Most bacteria are between 1 micrometer and 10 micrometers. These you can see with a light microscope. These you can see with the electron microscope. Electron microscopes, you can see much smaller things. We do not have them in lab. Light microscopes we do have in lab. So you'll be able to see chloroplasts and bacteria and plant and animal cells in the lab. So plant and animal cells are 10 micrometer to 100 micrometer. Butterfly egg is the size of that dot. It's about 1 millimeter. Butterfly, a centimeter or more. And then you can see all these things with the unaided eye. Humans were 1 meter or so tall. Say we should be a bit bigger than a meter. Probably like 160 to 160 to 200 centimeters. Blue whales, they can be about 80 meters long. California redwood, 100 meters. And that's pretty much that. And a kilometer is 4 laps around a track. So here we can see the surface area to volume ratio, like I was saying before with the cells. So if you have a 1 millimeter by 1 millimeter cube, so it's 6 sides times 1 squared equals 6 millimeters cubed. Its volume is 1 millimeter cubed because it's just 1 in each direction. So its surface area to volume ratio is 6 to 1. Okay, so let's make a 2 millimeter cube. So we have 24 millimeters squared for the surface area and 8 millimeters cubed for the volume. Now you have a 3 to 1. And you can see that we, just by doubling the size each time, we are having the surface area to volume ratio. So we have 6 to 1 here, 3 to 1 here, and 1.5 to 1 here. So each of these is half of the one before when the size of the cube doubles. So as cells grow larger, metabolic activity and need for resources and rate of waste production increases faster than the surface area. Some large cells increase surface area by folds in the cell membrane. So your intestines, for example, use this. So they'll be like this. That's what the cells will look like. Well, it's really only on one side, but whatever. So they'll have cilia, things like this, to increase the surface area to volume ratio. Some large cells increase surface area by folds in the cell membrane. So that's what this is. You'll see later in mitochondria, they have an outer membrane, but inside they have cristae. And looks like this. And they have all those folds to increase surface area to go across. To see small cells, there are 2 types of microscopes. There's light microscopes, which use a glass lens and light. The resolution is 0.2 micrometers. Electron microscopes. These use electromagnets and they focus an electron beam. The resolution is 2 nanometers. So you have your meter, and this is 10 to the negative 9 equals a nanometer. And so for a micrometer, you have your 1 meter times 10 to the negative 6 for your micrometer. So this is 1,000 times smaller. So research tools. So here we have our light microscope, and you can see cells easily. Here they're looking at plant divisions using mitosis. Here we have the scanning electron microscope, and you can see, if you zoom in enough, the phospholipids in the membranes. So chemical analysis of cells involves breaking them open to make a cell-free extract. The composition and chemical reactions of the extract can be examined. Properties of the cell-free extract are the same as those inside the cell. Cell structures and macromolecules can be separated according to size in a centrifuge. So you put it into the centrifuge with all its rings around the sides, and it spins around and it separates it out so that larger organelles and like phospholipid stuff that has been lysed will all be at the bottom. So this is your big stuff, and then you'll have tinier things like lysosomes, proteins, and things like that, and then you'll have much smaller things on top, and then you'll just have a layer of liquid on top of that. So it goes from bigger to smaller as you centrifuge it. So they can see the ratio of membranes, proteins, organic molecules, and fluid that is in a cell. So here is a better picture of the centrifuge. So you see nuclei, mitochondria, and endoplasmic reticulum. So this didn't separate the organelles. So the cell membrane is a selectively permeable barrier that allows cells to maintain a stable internal environment. So the cell membrane is called a phospholipid bilayer some of the time. It is important in communication and receiving signals. It often has proteins for binding and adhering adjacent cells. So it's basically just the bilayer of the phospholipids with the heads and the tails on the inside. So remember, a phospholipid looks like this: hydrophilic head and two hydrophobic tails. Okay, but on top of that, it has like proteins that hang out on or in it, and some go all the way through it. And then there's stuff that hangs out on top. So there's an extracellular and intracellular matrix. So there's structural molecules out here that help hold things together, and then there's signal molecules out here that can dock things and identify them and take in signals and stuff like that. So the cell membrane is really important. Let me open my drink. So there are two types of cells. There are prokaryotes, which have no membrane enclosed compartments. So karyotes equals nucleus. Pro means before. So this just means before nucleus. Eukaryotes, EU, means true in Latin. So eukaryotes just means true nucleus. So they have membrane-enclosed compartments called organelles. So organelles, super important. That was part of our levels of organization where we went from atoms to all the way up to the biosphere. Oops, okay. Ran out of room there. So organelles are made up of biomolecules, which are made up of molecules, which are made up of atoms. So it's about right here on our organizational levels. So organelles are just things like nucleus, chloroplasts, mitochondria. Prokaryotes do not have organelles. Eukaryotes do. So a prokaryote just has the one membrane on the outside. Eukaryotes have multiple membranes. They have the membrane on the outside, the membrane around the nucleus, and then membranes around all kinds of things in the cell, like the endoplasmic reticulum, the mitochondria, chloroplasts, vacuoles, Golgi apparatuses. These are all membrane bound. So prokaryotic cells are enclosed by a cell membrane, and they have DNA located in the nucleoid region. So you have your cell membrane, I hate how it won't let me write in this section. And then the nucleoid is just the area where DNA can be found. Nucleoid, okay. So the rest of the cytoplasm consists of cytosol. So the cytoplasm is everything inside the cell. So the cytosol is the water in dissolved material and suspended particles. And then there are also ribosomes, which are the sites of protein synthesis. So you'll just see dots as well inside of prokaryotic cells. These little dots are the ribosomes. So here we have a picture of a prokaryote. You can see the capsule, which is just a peptidoglycan coat with the cell membrane. So this one has an outer membrane, peptidoglycan, and then the cell membrane, the inner cell membrane. And these are all, the peptidoglycan and outer membrane are considered part of a cell wall. Prokaryotes and plants have cell walls. So then they have the nucleoid, which is where their DNA is. So they don't have a nucleus, they have a nucleoid. Then the ribosomes are the little dots. The cytoplasm is everything in there, and then the capsule is just around that, which is just protective. Most prokaryotes have a rigid cell wall outside the cell membrane. Bacterial cell walls contain peptidoglycans, which is just a carbohydrate. So whenever you see gly, that usually means it's a carbohydrate. And when you see peptid, you kind of can see, oh, there's protein in it too. So bacterial cell walls contain peptidoglycans, so it's mostly carbohydrate with some proteins in it. Some bacteria have an additional outer membrane that is very permeable. Other bacteria have a slimy layer of polysaccharides, so that's just another name for carbohydrate. So we can remember sugars equals saccharides, and carbohydrate equals polysaccharides. And this is called the capsule. Some bacteria, including cyanobacteria, have an internal membrane system that contains molecules needed for photosynthesis. So in cyanobacteria, they have these stacks that help them do photosynthesis so that they can get light waves. It looks a lot like chloroplasts. Some prokaryotes swim by means of flagella, made of the protein flagellin. A motor protein anchored to the cell membrane or outer membrane spins each flagellum and drives the cell. So here's your bacterial cell. There's a motor protein here, which really does look like a motor, which anchors the flagellum and rotates it so that the bacteria can be propelled. So here is the flagella. Some have multiple, some only have some, and then these small ones are called cilia. They can also be moved for movement, but it's kind of like using flippers rather than a tail. So here's that motor, really zoomed in. Here's a scanning electron microscope picture of that motor. So you don't need to remember all this. You just need to know that it has a protein that goes through the cell wall that acts as the motor. It has a C-ring, a transport apparatus, a rotor. So it's literally like a motor there. A P-ring, a drive shaft, an L-ring, and a filament. And this goes through the cell membrane, peptidoglycan, and outer membrane. So the cytoskeleton, some rod-shaped bacteria have a network of helical actin-like protein structures to help maintain their shape. So the cytoskeleton. So it's the skeleton in the cytoplasm. That's one word. I didn't make it look like one word, but it is one word. So in the cell, they'll have all these things that help give it support and keep its shape so it doesn't end up like a goo. Eukaryotic cells have a cell membrane, cytoplasm, and ribosomes. as well as membrane enclosed compartments called organelles. So organelles are membrane enclosed compartments. So that means they each have their own little phospholipid bilayer around them. So inside the cell, there's lots of little membrane enclosed things. The nucleus, the endoplasmic reticulum, Golgi apparatus, these are all enclosed by membranes. Each organelle plays a specific role in the cell. Organelles are like little organs. So organelle is to cell as organ is to organism. So they are subunits that carry out a specific function. So they have a specific role. Okay. Sorry, my phone is ringing. So here's the mitochondria. Remember, there's a meme about it. It's the powerhouse of the cell. Your cytoskeleton is your cell skeleton. You have your nuclear pores, which just allow things to go in and out of the nucleus. The nucleolus, where ribosomes are made. The nucleus, that's where our DNA is. The rough endoplasmic reticulum, it's just kind of like roadways for your cells. It's just dotted with ribosomes. The smooth endoplasmic reticulum has no ribosomes. It's just for movement of stuff in your cell. Then you have your cell membrane, which keeps things out and lets other things go in. So it acts as like a border wall. Well, not a wall, because I don't want to call it a wall, because then you think it's a cell wall. It's not a cell wall. So it's a border stop point. Border filtering. That's a good way to describe it. Then your Golgi apparatus is kind of like your cell's post office. It chooses what stays in the cell, what gets shipped out. So it's your post office. And then you have your ribosomes, which are where proteins are made. I don't even know why I added that either. Okay, proteins are made. Centrioles, these are made up of microtubules, and they're for cell division. Free ribosomes, where proteins are made, but that are free floating. And then the peroxisome is for breaking down toxins. So that's an animal cell for you.